Our next panel is panel 3, Classwide Scheduling, the Effect on Scientific Research and Therapeutic Breakthroughs. And with that, I'll pass it along to Geoffrey Laredo, our moderator.
Hi, good morning, everyone from beautiful California. It looks, Yana or Holly, like you need to enable my own video. For folks, there we go. And I apologize for a little bit of glare behind me. I'll try to turn myself around. Well, maybe that's a little better. I've been having some technical difficulties this morning, so I will apologize to everyone ahead of time if I am a little glitchy. For any of the hosts, if this remains too glitchy, please feel free to cut me off and let the panelists do their thing. Thank you, everyone, for joining us today. I've really appreciated the remarks from all of the previous panels. My role here today as a moderator is mostly just to spend just a minute setting the stage for this topic and then to get out of the way. I'm joined today by Dr. Sandy Comer of Columbia University and Dr. Patrick Beardsley from Virginia Commonwealth University, and I'll let them introduce themselves in just a couple of minutes. In my little bio, if you've chosen to read it, you see that I spent a lot of years working in the addiction research field at the NIH, both at NIDA and NIAAA, a lot of years. I am a 30-year, mostly retired Fed for my sins. So I bring that both small b and capital B bureaucratic experience to this discussion. For several years before leaving NIDA in 2018, some slice of the Schedule 1 issue was always on the policy agenda. You've already heard today, and you probably realize this because you're attending this conference in the first place. It is a critical issue. It's been a critical issue, and it's always been some kind of a flashpoint, whether we like that or not. And for any of you there who know me, you know that I have a lot of opinions about this. Today, though, I'm moderating this particular session, so I'll do my best to stay focused. Bottom line for me, from my years at NIDA, working with the research community, is that any class-wide scheduling is going to hurt research even more than Schedule 1 already hurts the research enterprise. We could spend the rest of the day talking about any even little slice of research problems due to Schedule 1. This is a big deal. So let me let you hang right there. I'll turn this over to Sandy Comer, who's going to provide us with a short slide presentation where everybody can geek out on science for just a few minutes. And then Patrick will add his thoughts and perspectives. Depending on the time available, I do have some questions that I might ask the panelists. We encourage you to put questions into the Q&A. And like I said before, the hosts have been really helpful to me. And if my own tech fails us, they will step in and help us finish this session. So Sandy, I'll ask you to take it away.
Share my screen. Okay, so my name is Sandy Comer. I'm a professor of neurobiology in the psychiatry department at Columbia University. And my background is in more basic kinds of research than what we've heard up until now. So I love data, so I'm going to share some data with you. And the topic of this panel is why we as scientists are concerned about class-wide scheduling. The first reason is that chemistry doesn't predict pharmacology, and I'll explain what I mean by that in just a minute. And then I guess more importantly in some ways is that this type of scheduling action could have pretty profound impacts on research and create barriers to developing medications for treating opioid use disorder. So we've already talked about, kind of what fentanyl is. I just want to reiterate Dr. Marino's point in the last panel that, fentanyl is a very useful medication clinically. It's used routinely in the operating rooms as a general anesthetic. It's available in various formulations for treating, breakthrough pain, for example. And it's not the, pharmaceutical fentanyls that we're talking about here. It's the illicitly manufactured fentanyl. It can be synthesized from this substance, N-phenylethyl 4-pipridone, which is a very inexpensive substance. And through a couple of very easy steps, it can be used to create fentanyl and carfentanil. So there's a very large profit margin for street chemists to make these substances. In terms of its pharmacology, so fentanyl differs from heroin, which is rapidly metabolized to morphine, in a couple of really important ways. So, both of these substances work at mu-opioid receptors. There are a couple of sites that are important for their pharmacological action. Morphine produces effects preferentially at the G protein site, whereas fentanyl has greater activity at the beta-arrestin site. And, in terms of how it differs from a pharmacology perspective, fentanyl is more potent. We've already discussed that. It's more lipophilic and it crosses into the brain much more easily. What we're really concerned about here from a public health perspective, as we've described, discussed earlier, is the propensity for fentanyl and its analogs to produce overdose. So these are some figures of fentanyl in the top, alfentanil, which is an analog, and remifentanil. So each line represents a different dose of the substance, and this is a measure of its effects over time. So all of these substances can produce very rapid and profound decreases in respiration. So this is 10 minutes here, where you get these really large decreases in breathing. This is a study that was very recently published by Sean Flynn in Charles, France, showing that carfentanil, fentanyl, and heroin produce effects that are very similar. This is a procedure that is used to kind of model, drug effects. And so all of them produce very similar levels of effect in this particular model. This is a dose effect curve here, so different doses along the bottom. Carfentanil at much smaller doses will produce the same effect as heroin. And then down here, what I'm showing you, we just talked about fentanyl having no legs. This is what I think you're talking about. This is the time course of the effect here. Fentanyl is very short acting. It lasts for, maybe an hour and a half. So Dan is absolutely correct in that regard. Whereas carfentanil produces a much longer lasting effect. So this figure using that same model shows that an opiate antagonist, naltrexone, which is currently approved by the FDA in various formulations for treating opioid use disorder. So what I'm showing you here is that the same dose of naltrexone that produces this pretty large blockade of fentanyl's effects is producing about a similar level of blockade for heroin. What's concerning is that this same dose of naltrexone is not blocking the effects of carfentanil quite as much. And this is a concern for naltrexone as a maintenance medication, but it's even more of a concern with naloxone, which is the drug or medication that's used to treat opioid overdose. And we've seen in recent years an increasing number of clinical reports demonstrating that higher doses of naloxone may be required for opioid overdose, and especially against carfentanil. And the reason why this may be happening is that there's a non-opioid component of carfentanil and fentanyl-related overdoses. So what they do that differs from some other opioid agonists is that they have a high propensity to produced a chest wall rigidity. And so this is not really reversed by naloxone because it doesn't act on these noradrenergic receptors. So as I mentioned earlier, the reason that we're concerned about class-wide scheduling is that chemistry doesn't predict pharmacology. And very small changes to a chemical structure can make a potent agonist into an antagonist. So an agonist is something like heroin or fentanyl, and an antagonist something like naltrexone or naloxone. And this is a perfect example of that. So the structure in red is the same across all of these different substances. This is oxymorphone, which is a very selective, potent, highly abusable opioid. And these tiny little modifications to this basic structure turns it into naltrexone or naloxone, which have saved, millions of lives. This is from a very recent paper by Ivy Carroll, a medicinal chemist, showing the same thing. So this is the structure of fentanyl, and this is a fentanyl antagonist. So it's very small modification that turns this into this that potentially could be very effective, maybe more effective than naloxone in reversing opioid overdose. And you would think that, something that shares a chemical similarity would be something that would be more effective in reversing its effects. In terms of legitimate, medication development, this is another example. This is a compound mirfentanil, which was under development for, for various indications, including as a potential maintenance medication for treating opioid use disorder. I received these slides from my colleague, Chris Cunningham, who's a medicinal chemist, and he wrote that, as a chemist, he would have expected mirfentanil to have high abuse liability, but Dr. Charles France, the author of that last study that I showed you, did some pharmacology studies showing that, mirfentanil may not have as high abuse potential as, as something like fentanyl. So this compound shares the general structure of fentanyl, and this would be captured as a Schedule I substance under the current class-wide fentanyl scheduling order. Another area of research that could be, impacted by the scheduling action is the development of vaccines. I'm working with my colleague, Dr. Marco Pravatoni at the University of Minnesota, who has developed a variety of vaccines, including oxycodone, heroin, fentanyl vaccines. There are other investigators across the country, Kim Janda, Gary Matias, who've also developed vaccines. And the thing with vaccine development is that it has multiple components that basically have to be put together, kind of like a puzzle. And there are different manufacturing sites that will do that. So not only would a Schedule I license be required by someone like Dr. Pravatoni, who developed these medications, but they would be required by the contract manufacturing organizations that would put the vaccine together. So, and this is what, something that Dr. Pravatoni experienced, and it took months and months for, first the CMO to agree to get a Schedule I license. I mean, there aren't very many facilities in the US that can do this type of work. And, willing to spend the money in order to, meet the requirements for the Schedule I licensing. In addition to, those kinds of practical problems associated with putting substances into Schedule I, there are other, more intangible problems as well, because this could, this type of action could disproportionately affect younger investigators who may not have the luxury of having the collaborations with senior investigators who know how to navigate the process, or investigators from smaller institutions that may not be willing to allow Schedule I type research to be conducted at their facility because of of all kinds of regulations that are required. And Chris Cunningham the medicinal chemist that I mentioned before is from Concordia University, and this is something that he's encountered. So what do we need? We need a mechanism for differentiating agonist versus antagonist activity, and we need exemptions from Schedule I requirements for those conducting research with Schedule I compounds. So my colleagues and I have published this paper very recently in Drug and Alcohol Dependence, and this, just basically outlines what I just told you. Again, I want to highlight my co-authors on this paper, Dr. Pravatoni, Andy Koop is also a medicinal chemist, Chris Cunningham, and Mike Bauman from NIDA. And I'd also like to acknowledge the CPDD Public Policy Committee, as well as Ed Long and Katie Van Lendingham from Van Scoyoc Associates. So, thank you.
Thanks, Andy, very much. Patrick, can I ask you to make some remarks?
Well, thank you, Jeff. I'm a professor of pharmacology at the Virginia Commonwealth University, and I've been involved with the study of drugs of abuse for over 30 years, both in terms of evaluating their abuse liability properties and for the development of medications for treating drug abuse problems. I'd like to make a few comments and observations, some of which Dr. Comer didn't mention, and I would like to expand on one she did mention. My comments won't only be restricted to the effects on research by class-wide scheduling of the fentanyls. Yes, I'm a researcher, but I'm also on a committee for the World Health Organization, which makes regulatory recommendations for the international scheduling of drugs under the United Nations Drug Conventions. So I view this present topic today both from the eyes of a researcher as well as someone who participates in the regulatory process. As a consequence, I have a few comments as a researcher, but I also have a few comments through a regulatory eye. My first comment is that it appears that history indicates that the DEA is able to keep up with the fentanyls following the current process under the Controlled Substances Act. 35 fentanyls have already been permanently scheduled under the current process. 18 of those fentanyls have been scheduled just in the last 2 years. Just this month, 10 additional new fentanyls were proposed by the DEA for permanent scheduling. Once those become permanently scheduled, there will have been 45 fentanyls permanently scheduled under the current process. And here, I'm not even mentioning the fact that all fentanyl-related substances have been placed under temporary Schedule I regulations since 2018. And it's relatively easy for the DEA to place a compound under temporary Schedule I conditions. All it takes to do so would be for a couple of in vitro tests, maybe a couple of laboratory animal tests, and some evidence of the scope, duration, and significance of abuse of a compound. Temporary scheduling of a compound lasts for 2 years. That can be extended for an additional year. Adequate time to gather evidence for permanent scheduling of a compound. The bottom line here is that history and data indicate that the DEA can keep up with the fentanyls without needing new powers. The second comment I would like to make is that empowering the DEA with unilateral powers to schedule an entire chemical class of compounds without input from the scientific and medical community, namely HHS, is inconsistent with international practice and with our current governmental philosophy. It is inconsistent with international scheduling because that process, as described under the Drug Conventions of the United Nations, starts with the World Health Organization. The WHO has a committee that conducts an intense scientific and medical review of a drug before a recommendation for scheduling is made to the United Nations. I'm quite familiar with this process, for I've been serving on this committee for several years. It is important to note that American scientists and representatives played a major role in designing this process when the United Nations Drug Conventions were written. Empowering the DEA with unilateral powers to schedule an entire class of compounds without scientific and medical input on individual compounds is historically inconsistent with what America advised for the international scheduling of drugs and how drugs are presently internationally scheduled. Secondly, turning over the power to schedule an entire chemical class of compounds to the Justice Department without input from the HHS also seems inconsistent with the current philosophy of the present administration that has repeatedly emphasized that science and data directs policy regarding the health and welfare of Americans. Class-wide scheduling of drugs would send a signal to the international community that science isn't necessary for determining health-related policy in the United States, and that policy can be solely determined through law enforcement officers and bureaucrats. A third point I would like to make is an expansion on a comment that Dr. Comer made. Research will be stifled by the permanent class-wide scheduling of fentanyls. In my past, I worked for 2 different pharmaceutical companies. I would find it doubtful that the pharmaceutical companies would be willing to explore the fentanyls for possible therapeutics that begin having a stain of Schedule I on them, and that incur the associated bureaucracy and security controls that are associated with handling them. It should be remembered that Paul Janssen of Janssen Pharmaceuticals originally developed the fentanyls because he saw opportunities to develop analgesics without the relative toxicity of morphine-like compounds. And there is definite hope in that regard. Dr. Comer showed us a couple, but also, for example, University of Maryland scientists have identified a fentanyl-based compound that is a potent opioid analgesic, but appears to have minimal respiratory depressant effects. If a drug like that could be validated and refined, it would be a major breakthrough in healthcare because opiate-like analgesics kill by their respiratory depressant effects. Those same scientists also identified a fentanyl-based antagonist, that is an antidote, as powerful as naloxone for reversing opioid activity. This also could be a major breakthrough if pursued. Naloxone is currently our go-to drug to treat opiate overdose. Naloxone has a chemical structure similar to morphine and heroin and is effective against those drugs. Naloxone isn't as effective against the fentanyls. Perhaps having an antidote based upon fentanyl structure would be more effective in reversing overdose by the fentanyls. However, it's very unlikely that there will be interest by pharmaceutical companies in further exploring fentanyl-related substances as safer analgesics or as antidotes to fentanyl overdose if the entire chemical class of fentanyls were placed under the strictest regulatory control of Schedule I. Of Schedule I. A related concern is the difficulty of moving a compound from Schedule I once it is placed in there into any other less restrictive schedule that is appropriate for a therapeutic. For example, only one drug has been moved from an initial Schedule I classification into a less restrictive schedule in the last 27 years. The fourth point I would like to make is really a fear that I have. Empowering the DEA to permanently schedule the fentanyl class of compounds would be the tip of the iceberg. There are other emerging opiate-like drugs of abuse that don't share in fentanyl structure. For instance, there are drugs based upon the drug adenosine structure that have been referred to as the nitazines that have recently come to be abused. Once all fentanyl-related substances are permanently scheduled, do we then schedule the entire chemical class of nitazines? And will it stop with the nitazines? I would like to end with a personal anecdote how class-wide scheduling of fentanyls can impede spontaneity in research and in research collaborations. I am working with a chemical engineer who has this vision that he can create a patch, much like the nicotine patch, that can sense the presence of fentanyls in the body and immediately release opioid antidotes. We have a small internal grant for developing this patch, but our collaboration has been hampered by the fact that although I am approved for handling fentanyls, he is still awaiting approval for his registrations. Obtaining a Schedule I registration can take a year or more, and he can't conduct research with the fentanyls because he is still awaiting is registrations. So in summary, first there appears the DEA is presently keeping up with the fentanyl story under the current process, and empowering it with new powers appears unnecessary. Secondly, allowing the Justice Department to schedule an entire chemical class of drugs is inconsistent with how drugs are scheduled at the international level, as well as the present administration's emphasis on science and data driving the health policy for Americans. Third, research will be stifled, especially pharmaceutical research, once an entire chemical class of compounds is permanently scheduled. And fourthly, I fear if the fentanyls as a chemical class are permanently scheduled, it will open the door to unbridled scheduling of other chemical classes of compounds, possibly hiding breakthrough therapeutics into a process that may never stop. Thank you.
Thanks very much, Patrick. So what I'd like to do is first say, Sandy, thank you for your presentation and giving us a little bit of the science background. Patrick, what I want to say from a science policy perspective to our audience here is everything he said. Take notes on every single thing he said. Watch the recording. That's your argument. I think this is an illustration of why I enjoyed my job at NIDA so much, working with folks like both of you. One comment from me and then some follow-up questions from me and the audience. When I was at NIDA, I worked in the policy office. I'm not a scientist. I was the interface with the Congress, and I'm a public health guy and reasonably knowledgeable on addiction science. And it was my job to help translate a lot of what folks that you've all heard and seen today have been working on, and lots of others across the field. I was told to my face by a senior DEA intervention policy person that I personally and my colleagues at NIDA were aiding and abetting drug dealers by opposing the DEA's efforts to somehow regulate the fentanyls more than they were being regulated. This was a few years ago, so it was before the most current attempts at class-wide scheduling, but it was basically the same discussion. So I wanted to just throw that out there and let that percolate into your brain, because that is A, unacceptable, B, incorrect, C, completely crazy for a federal agency staffer to say to another federal agency staffer when you're allegedly trying to figure out difficult things. We know that the research community and the Drug Enforcement Administration are going to find plenty of issues to argue about. That's okay, but argue about them constructively as best you can. But I just wanted to throw that out there because it was an experience that I will never, ever forget. It was all I could do to maintain my own personal calm. Again, for those of you who know me, I don't take well to things like that, but it really shows the toxicity of some parts of this discussion. So I'd like to ask a couple of questions first of my own of both Patrick and Sandy. Are there, in the work that you've done yourself, have there been specific requirements of any of the Schedule I issues that have caused you difficulty in your work? From previous discussions that we've had, your experiences have been a little bit different, and I think it's valuable for the audience to hear that. So whomever wants to go first.
I could comment on that, Jeff. Okay.
Sure.
Right. Well, the present situation is really impossible for researchers. That is, there is this class-wide temporary scheduling of all fentanyl-related compounds. Okay. Along with that, individual fentanyl compounds get permanently scheduled. So when in 2018, when all fentanyl-related compounds were temporarily scheduled, I went through a very intensive process of applying for permission to work with fentanyl analogues. And I was granted permission. And I obtained several of these fentanyl analogs to work with in my laboratory. In fact,
How long did it take for you to get that permission? Was it a drawn out thing or did it work well for you?
I'm an atypical case, Jeff. I'll tell you why. Because at the time I had 2 contracts with the DEA to evaluate the pharmacological properties of the fentanyls.
Fair enough.
So I'm not sure if the speed for me to get approved for working with all fentanyls is representative or not.
Yeah, you're a ringer. Okay.
Yeah. In any case, the important point is that there is a very intensive process of getting approved to work with all fentanyl related substances. And I went through that. But now when some of these compounds that I brought into my lab and have done research with under the auspices of that permission have now become individually permanently scheduled. And I have to go through the process yet again for gaining permission to work with these individual fentanyls that have become permanently scheduled. In fact, I probably am holding some fentanyls illegally because I initially brought them under the auspices of all fentanyl related substances, but now some of these compounds have become individually scheduled. And it makes, it's just a bureaucratic mess and just enormous hurdles to keep up with the paperwork to do research with these fentanyls under the current situation.
Thank you. Sandy, do you have anything that you could add to that?
Yeah, I guess I just want to thank you for sharing the anecdote with your experience with the DEA agent. And just, I just want to, I guess maybe since we're being recorded, I want to say that we're all on the same team. The DEA, the scientists, we recognize that this is a serious public health, issue. We're all incredibly concerned about it. And, we're just working at it from different angles. We as scientists are working so hard trying to find an effective, antagonist for reversing overdoses to help, help when in those situations where naloxone's not working. We're looking for vaccines that might, be useful both for people who have opioid use disorder and also first responders who may be exposed to these substances in their line of work. And the one is stymieing the other, and we have to figure out a way to make it work for everyone. So I'll just say that off the at the beginning. So in terms of my own research, the example that I gave with the vaccine development is probably the closest, where because I'm testing Marco's vaccine right now, and there was a delay for about a year before the CMO was able to get a Schedule I license and then get everything in place regulatory wise in order to develop the vaccine. So yeah, I've been very directly affected by this. There have been other investigators that I know in the field who have who've had similar problems, and some of the very common themes that we've heard from the scientific community is the very long time frame until initial approval, which is compounded by both state and federal licensing requirements. There, as Patrick mentioned, there are delays with adding drugs to an existing license. There are communication difficulties after you submit your application. Sometimes people have told me that it feels like their application got lost in a black hole. They can't find anybody who can answer their question about where their application is in the process. There are practical difficulties and hurdles with regard to storage requirements. So these are not insignificant hurdles. These, storage requirements are expensive. And, a lot of us, researchers are funded by NIDA, and we can't, build those kinds of costs into our grant applications. So, these are very real hurdles that we're facing.
Thank you. And I appreciate we're all on the same team comment, and that's exactly true. And I do think even with the process or bureaucratic hurdles that researchers have faced, it's my personal sense that in tone, if nothing else, things have improved over time. NIDA and DEA were actually invited by CPDD a couple of years ago to have a large joint session speaking directly about this issue to try to solicit more input from the research community. At the beginning, at least in my personal experience, at the beginning of this argument, both of you have talked about, some things taking a long time, some things not taking a long time. One of the dirty little not so secrets at the NIH, specifically on this topic, when I was there, is many of you in the audience might know that many NIH Institute directors have labs of their own, either at the NIH or elsewhere, whatever the arrangement they've made with the NIH. Well, NIDA director, Dr. Nora Volkoff, of course, has had a lab for all of her years, for all the years that she's been the director at NIDA. And she wanted to do research and get her lab to do research specifically on these fentanyls, starting a few years ago when it was becoming an even bigger problem for the country. And without getting into too many of the details, I mean, think about this. Here's an NIH Institute director whose job it is to direct $1 billion agency to try to help figure out this problem, who has a lab of her own that she directs, who is saying, hey, I'd like to personally try even more to do some of this work. That lab, they had not sought a Schedule I license prior to this, and it took that lab over a year to get licensed. So the bureaucratic hurdles are real. I don't have to be nasty about it, but it's been just process-wise an extraordinary challenge for a lot of hardworking folks to try to figure out what to do. We have a couple of questions in the Q&A that I'll try to get to here. The first, if either of you have any comment on this, is from Christine Leonard, and I'll just read it to you. There have been reports that xylazine mixed with fentanyl has resulted in a significant number of fatal opioid overdoses in Philadelphia. Can you comment on this issue and the use of conventional overdose strategies for these types of situations? Does either of you have anything on that? It's not exactly on our topic, but I didn't want to just ignore your question, Christine. We can certainly try to get you an answer separately from this panel if neither of our panelists have that.
Yeah, let's do that. I'll definitely look it up and get back.
That's an unusual drug combination.
Fair enough. Okay, Christine, we'll do our best to get back with you on that. There's a comment in the Q&A from Heather Harris that says the DEA is a law enforcement agency, not a scientific one. I'd like to make a first comment on that. I agree with you. I think that is 100% correct. The problem is that there is a huge chunk of what the DEA does where they actually are supposed to be, whether we all like it or not or agree with it or not, they are supposed to be a scientific agency. They do a whole range of testing. They have the legal authority to regulate pharmaceuticals. So by law, they actually are supposed to be a scientific agency. Lots of folks out there in the science policy world have argued for years that, gee, maybe that really shouldn't be their job. Let's put that kind of work where it belongs in the Department of Health and Human Services, and let the DEA, in fact, be a law enforcement agency. I'll leave it at that. I don't know if either Sandy or Patrick want to extend that comment.
Well, I could comment, Jeff. Actually, the DEA does have a scientific staff that reviews drugs during the scheduling process. I've come to know some of these, and they actually are very good scientists. At least in their past, they may have been scientists. And so there is an aspect of the DEA that is equipped to do some scientific review.
Yeah, and I guess I just to add to that, you know, like one of my concluding points was that we really need to come up with a system for differentiating an agonist for an antagonist. You know, obviously, the definition of a Schedule I compound is that it has high abuse potential and no therapeutic use. So, if you follow this class-wide scheduling and sweep up all of these substances, some which may not have any agonist activity at all, you're not meeting the definition of a Schedule I compound. So, from a very basic level, it's I think it's crucial to identify what's an agonist and what's an antagonist. And this process that Patrick is talking about is something that the DEA is currently using to characterize these new fentanyl-like drugs. So that process is already in place. And, one thing that we haven't talked about yet is the 8-factor analysis that is required to, place a substance permanently into Schedule I. And that, that's a very long, cumbersome process. But, what I was, suggesting that we do is use these really quick, simple kinds of procedures. Like, some of your procedures, Patrick, I know are not simple, but it's relatively rapid, a relatively rapid process to do this kind of separation of something that's an agonist versus an antagonist.
I think that's a really important point. And even just from an operational perspective, Sandy, you used the term sweep up, and that actually triggered a memory of mine. When the first part of my involvement in this issue was several years ago, when there was actually proposed legislation based on some work that the DEA did to permanently schedule 340, 350 alleged fentanyl compounds into Schedule I. And it caused a heck of a stir within HHS. And NIDA and the Food and Drug Administration and DEA spent months working together, not always happily, but we worked through things because we on the health side could not figure out what many of these compounds were. So there's an understandable and really challenging tension at the DEA between the scientific side and the law enforcement side. And I think that's, that is very real. And at the end of the day, those three agencies were only able to agree on a couple of dozen of the compounds as ticking enough of the boxes, like, do we really know what this is? Do we really know the chemical compounds? Is it, as best we can all really tell and agree, is this really dangerous? Does it even merit a scheduling discussion? So it points to the legitimate difficulty, at least under the current bureaucratic construct at the federal level, of how all of this gets dealt with.
Well, to circle back to the criminal justice, issue as well that we discussed this morning, you can imagine that with these, class-wide scheduling of these substances, sometimes, these drugs are found in trace amounts in other drugs. We talked about now they're being adulterated into cocaine or methamphetamine. And then the sentencing requirements are much harsher. You can imagine that if it happens that one of these substances was an antagonist, and but it meets the definition of a fentanyl analog, so it has no agonist activity, it would not get somebody high, then somebody could be sent to prison for years on something that doesn't even have any abuse liability at all. Right. Yeah. So there's so many unintended consequences of this type of scheduling action.
Yeah, and another challenge for all of you researchers, there's another comment actually in the Q&A from Dr. Marco Pravatoni that says one of the challenges is that each state, and Patrick touched on this a little bit as well, each state provides different guidance on acquiring a DEA Schedule 1 or 2 license. And so in his opinion, centralization would probably help researchers. And that's not the first time that somebody has said that to me. And it's not, again, it's not a matter of the DEA necessarily intentionally trying to make people's life difficult. It's just a difficult and cumbersome process that at the very least, it doesn't help the research community in its attempt to help provide some answers to a really long-term problem. So I don't want to artificially cut us off, but I see on the clock in front of my face here at 10:45. So I think I need to turn this back to the conference hosts. I want to thank you, Sandy and Patrick, for agreeing to be part of this. And thank you all very much for your attention.
Thank you.
Thanks so much, everyone. I have just been so excited this morning to see all of the different participants who have brought their expertise and viewpoints to this discussion. And we're not done yet. We have 2 more terrific panels coming up after a 15-minute break. So I hope you can stretch your legs, refresh your coffee, grab a bite to eat, and then come back and rejoin us at 2 o'clock when we're going to have Marilyn Mosby, who is the state's attorney for the city of Baltimore, sitting down with Liz Comar from Fair and Just Prosecution to talk about how this looks through her eyes. And then we're going to close out today's discussion with a terrific panel of advocates, including Sakira Cook, Jason Pye, Kanya Bennett, and Jasmine Bundella. So please take a break, take a minute, and rejoin us at 2 o'clock. Oh, yes, 2 o'clock.